afternoon, everyone. It's my very great pleasure to introduce Professor Linda Richards, who many of you will know, but some of you may not. Linda is a Professor of Neuroscience and Deputy Director of the Queensland Brain Institute at the University of Queensland. And Professor Richards' laboratory focuses on the development of the cerebral cortex and cerebral connectivity. In particular, as we'll hear today, the development of the corpus callosum. Last year, in 2015, Linda co-founded an international consortium for the corpus callosum and cerebral connectivity with colleagues from Melbourne, San Francisco, France and Brazil. This consortium brings together clinicians and scientists working to identify the causes of developmental brain disorders and how best to provide support and care for affected individuals and their families. Linda has received numerous awards and fellowships, and these include the Charles Judson Herrick Award from the American Association of Anatomists in 2004 and the Nina Kondelis Award from the Australasian Neuroscience Society in 2010. Since 2011, Professor Richards has been a Principal Research Fellow with the National Health and Medical Research Council. In 2015, she was elected as a Fellow of the Australian Academy of Science, and she's President-elect of the Australasian Neuroscience Society. She'll take up office in December this year. Just last week, actually, I noticed Linda's updated her qualifications, but Linda was one of 50 new fellows elected to the prestigious Australian Academy of Health and Medical Sciences. And Professor Richards, or Linda, as we know her, is passionate about the public awareness of science. In 2006, she founded the Australian Brain Bee Challenge. It's a competition for high school students to learn about the brain. And since 2006, over 30,000 students have participated in the Australian Brain Bee Challenge. This is a testament to Linda's energy and enthusiastic mentorship. Some of those students, in fact, quite frequently, students from Australia have won the International Brain Bee competition. Australian students have won the international competition 3 times, and they've been placed in every single event since the foundation of the Australian Brain Bee Challenge in 2006. So on that note, I'd like you to welcome Professor Linda Richards to present her talk, which is entitled Development of the Corpus Callosum.
Thanks very much, Jenny, and thank you also to Jeff and Trevor for the invitation to come, and I bring warm regards and greetings from my colleagues at the Queensland Brain Institute to all of our friends here in Melbourne. So my lab is really interested in how the brain is formed during development and how it's wired up correctly. And today I'd like to extend some of the really wonderful work that Mu-ming talked about in terms of synaptic plasticity, and to just float the idea of another type of plasticity in the brain that occurs during development, which is long-range axonal plasticity. So, of course, the presynaptic elements of the synapses are the axons, and they have to be wired and connected correctly in order for the brain to function properly. And collections of these axons form large fibre tracts over, that connect regions over long ranges. And the largest of these fibre tracts is the corpus callosum, which you can see here. It's so large you can see it very clearly by MRI. And the middle picture is an adult brain with the top part dissected off, so you're looking down on top of the corpus callosum. And the other image is a diffusion tensor tractography analysis based on diffusion imaging from our 7 Tesla MRI scanner. So another interesting fact about the corpus callosum is that it's only present in placental mammals. So marsupials and monotremes don't have a corpus callosum, as it evolved with placental mammals, even though marsupials have a 6-layered cortex. Their cortex is connected via another major commissure here called the anterior commissure. So there are people who are born without a corpus callosum, and we call this agenesis of the corpus callosum. And you can see here, these are the controls. This is a sagittal view, MRI view. And you can see this person who has absence of the corpus callosum, and this is a coronal view this way, and you can see the fibres, although they are generated up here in the grey matter where the cell bodies are, and they send their axons down towards the midline, and in these cases they're unable to cross the midline. Now, the symptoms of having agenesis of the corpus callosum vary widely, and part of the reason for that is that malformation of the corpus callosum occurs in over 300 different congenital syndromes, so that's where the person has a corpus callosum malformation but also another organ system deficit. And so the symptoms, as I say, range very widely from very high-functioning individuals who have, as the most common feature, social interaction problems. That's really the most common feature of the high-functioning individuals, to a more moderate group of individuals that have learning deficits and some sensory deficits, to very severe cases in syndromes like Aicardi syndrome, which is found in girls, where the girls don't learn to walk or talk, so they're quite intellectually and physically disabled. So we're very interested in understanding the genes underlying these disorders, but also how that relates to the wiring of the brain, and then how that wiring relates to the cognitive function of those individuals. So we use mouse as our model system. So I'll just introduce you to the corpus callosum in a mouse. So a mouse has what we call a lissencephalic or smooth cerebral cortex without Salcean gyri, and you can see labelled here are the cell bodies which will be sitting up here in layer 2-3 of the cerebral cortex, and we've labelled the primary somatosensory cortex here, and you can see that these cells send a projection down in between these structures, which I'll talk about a little bit later, called the barrels, and they arborise in layer 5, and then they send their projection across the midline to targets in the contralateral hemisphere. So the corpus callosum really connects very specific areas in the two hemispheres of the brain. So our lab has been investigating the mechanisms that regulate how these axons cross the midline. And more recently, we've been very interested in how these axons find their correct target in the contralateral hemisphere. So just going back to the midline guidance mechanisms, so shown here is a schematic of a neuron, a callosal neuron that would be projecting across the corpus callosum, and underneath is an image from the mouse brain, this midline region here. And you can see the callosal neurons, axons are labelled in red, and the glia that surround the tract at the midline, shown here, are labelled in green with an antibody against glial fibrillary acidic protein. So we know quite a bit about the formation of these midline glia here. They're regulated by a family of transcription factors called the nuclear factor 1 genes, which are absolutely essential for the differentiation of these glia. And I will come back to this a little bit later as I talk about some of our work on midline development. So in the field, mechanisms that regulate how axons cross the midline are highly conserved, all the way from flies and worms to fish and then humans. And we know a lot about the molecules that are involved. We know that on the growth cone of the axons are receptors for guidance cues, and these guidance cues are things like secreted proteins, which have names like these, SLIC2, semaphorins and netrins, and these are expressed by the glia, or tissues at the midline, and they help to guide the axons across the midline. So today I'd like to talk about another mechanism that's really important for formation of the corpus callosum that occurs prior to this guidance event. And this is work that has been done mostly by a postdoc in the lab, Dr. Ilan Gobius, and she's been working with a PhD student, Laura Morecambe. And for the students in the audience, I'd like to say that today I'm very excited to be able to tell you about this work because it is 7 years of work from Ilan. It took a long time to bring this whole thing together, but she has essentially uncovered the major mechanism that underlies complete agenesis of the corpus callosum. So I mentioned that people who have complete absence of the corpus callosum here, you can see that the axons fail to cross the midline. You can also see that there's a small structure that lies just underneath here. It's called the septum pellucidum. And we were very interested to understand how the septum develops. And you can see here that in the control person, you can see that the septum is really connected together and forms this bridge of tissue. And in a person with complete agenesis of the corpus callosum, you can see these septal leaves are quite open. And this will become very important as I take you through the data. So what I'm going to tell you about is how this region of the brain develops, and when it's open like that, there is a failure of callosal axons to cross the midline because they don't have a substrate to grow across. So in order to understand exactly what's happening here, we have to go back to very early development, and what you're looking at here is a mouse brain with looking at a frontal view by scanning electron microscope, and you can see very early in development, so these are embryonic days, and mouse gestation is around 19 days, so this is about just before halfway. And you can see that at this stage, the brain begins, the two talus phallic hemispheres actually begin as a single prosencephalon, which then grows out to form two talus phallic hemispheres here. And you can see that there's a large fissure in between the two hemispheres because they grow forward, and at the midline there is this large fissure. So although they're connected ventrally, they are not connected at the level in which the callosal axons will be crossing the midline. So a major remodelling event has to occur in order for the callosal axons to be able to cross. So to look at this very carefully, so this is some anatomical morphological data from mouse, you can see over here. So these are different developmental stages in the mouse, and here is one developmental stage in human fetal brain at gestational week 13, just to demonstrate that the same mechanisms are also occurring in developing human brain. It turns out that the mouse is an excellent model system for studying development of the corpus callosum because these early developmental events are highly conserved. So what you can see is that at early stages of development, the interhemispheric fissure is very deep, and there's a very small region of tissue connecting the two hemispheres together. We call this little tiny piece of tissue the hinge area, and what happens over development is that this region starts to remodel and fuse, providing what will become evident shortly, a bridge for callosal axons. So this is the corpus callosum here, to cross the midline. And the same thing occurs in developing human fetal brain as well. So you can see the fissure here, and here it's started to remodel. So this is in the same brain, and this process is really occurring at different levels, from ventral to dorsal and caudal to rostral. So in this kind of fashion, this is how the fissure is eliminated during development. So what is the cellular mechanism by which this is occurring? So glia, as I said before, are extremely important in guiding axons across the midline, but they're also incredibly essential for this remodelling event. So what you can see here are horizontal sections. So these are sections this way through the mouse brain. So you're looking down upon, the fissure is coming in, and you're looking down on top of that. And in a mouse, the septum is relatively large. So this is the septum here. This is the lateral ventricles on either side, and this is the third ventricle down here. So what we found is a population of cells in the third ventricle that actually express early glial markers such as nestin and also glast, but they don't express GFAP at these early stages. And these cells migrate forward and actually are involved in what we call zippering up the midline. So previously in the early 80s, Jerry Silver's group from Case Western had described glia at the midline that he called midline zipper glia that might be involved in some kind of fusion process. But he had not been able, at that time, the techniques weren't available for him to understand where the cells arose, how they did this zippering event, or whether to actually perform experiments to see whether to test his hypothesis. So it turns out that these cells do come from the floor of the third ventricle and appear to migrate forward. So as they start to migrate forward here, they actually change their morphology. So this again is the 13-week human brain, and this is a 14-week gestation human brain, and this is mouse brain shown here. So the fissure is of course filled with leptomeninges, so the meningeal coverings come down into the fissure, and this also has to be eliminated. And what you can see here are the shape of these cells is very bipolar, and then very quickly these glia begin to differentiate and become multipolar astrocytes, which produce processes that go across this cleft and actually begin to knit together the midline. And so Ilan actually used a technique called in utero electroporation, which I can describe a little bit later, but this is a way of specifically labelling those cells in the third ventricle with a glassed GFP plasmid, and she was able to show that these cells migrate forward and are bipolar in shape and then begin to change their shape to these multipolar astrocytes. So here is a developmental time course from embryonic day 12 through to E17, where you can see, again, this is looking at a horizontal slice, and you can see the fissure is very large here at this very early stages, and then the cells begin to proliferate at the edges here, and they actually push the cleft together so that the two pieces of tissue from either hemisphere come in close apposition. Then the glia begin to produce their processes across this cleft and start to knit together the midline. And you can see that this area is exactly where both the corpus callosum and another major commissure called the hippocampal commissure actually cross the midline. Now, is this actually required for corpus callosum axons to cross the midline? So previously, Sam Pleasure's group in San Francisco had shown some data that this area of tissue, the basement membrane here, expressed molecules which regulated the crossing of callosal axons, and they hypothesised that they could grow across this cleft even without having this remodelling event, that they could grow across this cleft depending on the expression of molecules. So we wanted to determine whether or not the axons actually use these glia or whether they were able to cross through the cleft. So in order to do that, we performed this electroporation technique. So this is a way to just specifically label the pioneering callosal axons that come from an area called the cingulate cortex, which is a very medial part of the cortex. And when we do that, so they're labelled in white here, and the midline zipper glia are labelled in red, and you can see that the axons grow down underneath the fissure and cross over where the glia are. In fact, in areas where the fissure is very deep, the axons will continue to grow down. So here they are growing down, and this is the inset from here, you can see this little pioneering axon coming all the way down in order to cross over where the intercalated midline zipper glia have grown. So they don't cross through the fissure or the leptomeninges. They have to cross over the glia. And you can see that here is an area where the fissure is being eliminated and the glia are forming this bridge, and labelled with GFP are some pioneering axons which are specifically crossing in this area. So the glia form these small pinching off areas across the fissure, which then fill in over time. Now, just incidentally, we have looked at marsupials and a platypus monotreme here, and interestingly, these animals don't undergo this remodelling event, so they don't have a substrate to form a corpus callosum. All right, so what are the molecular mechanisms involved in this cellular event? So previously, Professor Sue mentioned the fibroblast growth factor family, and this is a family of secreted proteins that are also very important for brain development. And this is an in situ hybridization of, for Fgf8, one of the FGF family members. And you can see Fgf8 is highly expressed at the floor of the third ventricle, exactly where these glia are originating from, and it remains expressed in this area. We've also looked at the expression of the receptors for FGF8, FGFR1, which is co-expressed on these glass-positive glia. And also you can see here by in situ hybridization, the FGFR1 is highly expressed in the area that's undergoing active remodelling. FGFR2 is also expressed in this area, as are other molecules that are involved in the FGF signalling pathway called the Sprouty or SPRY genes. So interestingly, it had previously been shown that FGF receptor GFAP-Cre, so these are conditional knockout mice. This is work from Flora Vaccarino's lab. She had shown that they have complete agenesis of the corpus callosum. And you can see, this is an image from their paper, that in fact they have this massive cleft that hasn't remodelled. And there are two human syndromes where FGF receptors are mutated, and these children also have deficits in the corpus callosum, mostly associated with septal defects. So we perform conditional knockout of FGF8 using a Neston-Cree driver line, and this creates a kind of not complete knockout, but we can't use a straight FGF8 knockout because FGF8 is also involved in cardiovascular development, so the animals die very early. And you can see that in the FGF8 conditional knockout, this is a milder phenotype than you would see with a different Cree driver line, that the fissure is retained and we get the formation of what we call Probst bundles. So these are sort of equivalent to what you see in humans with complete agenesis of the corpus callosum. These bundles were first identified by a person called Probst in 1901, so they're named after him. So we also performed gain of function experiments. So this is the in utero electroporation technique I was telling you about before. So this is where we can introduce a DNA plasmid into the brain of developing embryos in the mouse during development. It's a fantastic way of looking at cell migration or trying to specifically alter gene expression in a particular population without knocking it out in the whole brain. So we overexpressed FGF8, and you can see here's a single case of that where we've used an antibody against FGF8. So we have an overexpression and our control side here. And you can see that it's overexpressed in these glia. And when we do that, we get an upregulation of glial-associated genes, such as S100 beta, EAT1, which is also called GLAST, and nuclear factor 1. So this is the transcription factor family I was talking about before. And when we do that, you can see, depending on the age at which we do the electroporation, we get precocious glial differentiation, and this is inhibitory for fissure remodelling and formation of the corpus callosum. So to summarise that part, either loss of FGF8 or overexpression of FGF8 and precocious differentiation before, particularly in one side of the midline, prevents this fissure remodelling event from occurring and callosal axons from crossing the midline. So we've been looking at the downstream signalling pathways involved and what these could be. And it was known from the literature that MEK, intracellular signalling molecule, MEK1 and 2 knockout mice also have complete agenesis of the corpus callosum, and they have this fissure, and as well the NF1A and B knockout mice. This is work from our lab. So I'm just really summarising a lot of work here, and I can't go through everything, but what you can see is if we overexpress FGF8 in one hemisphere, we get an upregulation of GLAST and an upregulation of GFAP. If we do this in either an NF1A or NF1B knockout mouse, you can see that we get upregulation of GLAST, but we don't get upregulation of GFAP. So NF1A is required for the differentiation of glia mediated by FGF8. And we've also looked at human patients with NF1A and B haploinsufficiency, and many of these patients also have a retained interhemispheric fissure, and this is equivalent to what we see in the knockout mice. So just to conclude this part of the talk, FGF8 regulates NF1A signalling, which binds specifically to the GFAP promoter through a MEK-ERK signalling pathway. Now, I think I might just run through some work at the back here because this part's been published and I can see I'm running out of time. We previously have been very interested in how axons are able to find their target in the contralateral hemisphere, and the work I just skipped over was some work showing that activity is incredibly important for the targeting of callosal axons into the contralateral hemisphere. And so I just wanted to leave you with this, that although we see very severe cases of callosal agenesis such as these, we have evidence from that work that although callosal axons may be able to cross the midline, if they don't target correctly, then we also have targeting deficits just in the contralateral hemisphere. And so a question arises as to whether all brains are wired the same in terms of their axonal connectivity, and this is what the International Consortium is working on. And some of the groups in that consortium have shown some amazing axonal plasticity in people with partial agenesis of the corpus callosum, shown here, and that these subjects have very different axonal connectivity. And this work's also been replicated by one of our consortium members, Fernanda Tovar-Moll in Brazil. And we've also found ectopic tracks in ACC subjects. So just to conclude, long-range axonal plasticity occurs during development, and it could have implications for understanding intellectual disability, autism and mental illness if we consider not only the major structural changes that we can see by anatomical MRI, but if we can push the technology, the tractography mechanisms that we can, or techniques that we can use, we may be able to see more detail about how axons target in the contralateral hemisphere, and you can read more about that hypothesis in this article. And I'd like to acknowledge the people in my group who have done this work, in particular the work done by Ilan and Laura on the midline fusion. Thank you very much.
much, Linda. I think we've got time for one or two very quick questions. Has anyone got any questions for Linda? Hey, go ahead.
I just think of possible clinical applications of this, because usually within humans you're not going to find out that the corpus callosum is missing until they're at a certain age of the toddler or what.
Actually, you can see it by ultrasound at 20 weeks.
20 weeks.
Mm-hmm.
So that make it easier. So what possible therapeutic applications would you come up with now that you know that these base cells that create the structure and you know the growth factors that create them? So like a stem cell therapy that you could create?
Right, so I think that it's not possible to really start considering fetal surgery in order to repair the bridge. But what this work has been able to do is to identify the cellular mechanisms and therefore then the molecular mechanisms that are involved. So there is no genetic test that really can be applied at the moment for agenesis of the corpus callosum. So at 20 weeks, if you have a fetus with complete ACC, you don't know where on the spectrum that child might fall, and so having a genetic test is our first priority. But I guess in the future one could think about, but that would be fetal surgery.
Thank you, Linda. It's always puzzled me about the very large number of syndromes associated with agenesis and why it was useful to lump them as coming under that, and you've sort of explained this beautifully. Of the 300 odd syndromes, the genes are known for probably 100 or so.
70.
70. So when you look at the list, can you then sort of see a way that it all might be linked into this development of the septum and the fusions? Does it all kind of work to knit it all together? Yep.
So I talked about midline disorders today, but there's also axonal development and growth disorders. There are also secondary effects, like the Aicardi syndrome doesn't fall neatly into one of these because the girls have very large cysts at the midline, and this is a sort of disruptive to callosal formation rather than being cell autonomous, probably. So there are a huge number of things that can go wrong when the corpus callosum is forming, and that's the genes that are involved in each one of those steps do fall out in terms of these, the different mechanisms.
Okay, we've got 1 minute. Who wants to squeeze one in? Carolyn.
Sorry, thank you. That was fantastic. Can I ask, a lot of neurodegenerative diseases are associated with thinning of the corpus callosum and all those agents.
Yep.
So will you be thinking of thinning of the corpus callosum in the same breath as agenesis?
Yeah, so we're also interested in callosal hyperplasia. That's quite an interesting. So what we're looking at there is whether or not the patient has only thinning of the corpus callosum or loss of white matter throughout, and then it might be some more leukodystrophy. So how you get fewer axons crossing the midline. There's evidence in mice that if the neurons are not specified correctly, they may actually project subcortically through the internal capsule instead of projecting callosally. So it depends if the patient has Probst bundles. That kind of tells you, something. And then you've got all of the disorders where you have partial ACC as well. So that's kind of, just going back to Sam's question, that's kind of why everything is lumped in together at the moment. But there are, some quite distinct differences in the developmental mechanisms involved.
Okay, now we have to really quickly thank Linda for a really elegant and data-packed talk. You can catch up with her after the meeting to ask any more questions that you might have.
Well, good afternoon, everybody. My name is David Graydon, and I'm the Deputy Head of Electrical Engineering and the leader of the Bionics Lab at the Centre for Neural Engineering. It's my enormous pleasure to introduce Professor Mark Cook, who is currently the Sir John Eccles Chair of Medicine and Director of Clinical Neurosciences at St Vincent's Hospital, Melbourne, and he specialises in the treatment of epilepsy. After completing specialist training in Melbourne, he undertook an MD thesis at Queen Square, London, and then he returned to St Vincent's Hospital, Melbourne, where he developed techniques for the accurate measurement of hippocampal volumes and established their position in non-invasive assessment of surgical candidates. More recently, Mark's interests have included computational models of epilepsy, seizure prediction, and cortical excitability. His research has taken on a strong commercial focus over the last 8 years, mainly in the development of an implantable device to treat epilepsy and novel polymer-based drug delivery systems. He has an avid enthusiasm for gadgets and new technologies, which has led to some very interesting research, most recently in MEG and wearable devices. He's an author of 7 book chapters and over 200 published peer-reviewed original papers and abstracts. Mark has very strong collaborations, nationally and internationally, particularly with the Bionics Institute, Centre for Eye Research Australia, University of Wollongong, Institute of Neurology London, Royal Melbourne Hospital, Penn State University, and, most importantly, the Department of Electrical Engineering at the University of Melbourne. And, to top it all off, he's an all-round nice guy. So it's my pleasure to hand over to Professor Mark Cook, who will present on new insights into the dynamics of epilepsy and seizure prediction. New insights into the dynamics of epilepsy through long-term invasive monitoring. Thanks, Mark.
Thanks a lot, David. Thanks for the invitation, Trevor, and it's great to be able to present this work, but I want to thank David especially because our collaboration's been very strong over some years, and none of this obviously would have happened without that. But as well, he gives a good intro and said nice things. I was going to give him my own little blurb to say, but his was much better. So I was going to tell you a little bit about what we've learned from long-term monitoring as part of a study that we did and where that's led to. So I'll give you a little bit of background first and then talk about the chronic monitoring studies. And there have been other monitoring studies done chronically other than this one, but very limited, and really the only other genuinely long-term chronic invasive monitoring studies of epilepsy have been through studies like NeuroPace, which have been therapeutic studies that don't collect the same continuous data that we've had. But I'll talk a little bit about the insights that we've obtained into the patterns of epileptic activity, spikes and seizures, and some of the most interesting circadian rhythms, both in ultradian and infradian changes that we've seen throughout, and why this might be important in chronotherapeutics. As well, I'll talk a little bit about the future, some of the work that we've been doing around the computational analysis of this data, and as well some of the work about less invasive systems and how these might integrate with peripheral devices, which are becoming increasingly popular. Now, the invasive monitoring of this time has only really become feasible in recent times because of the challenges around the technology, particularly battery power and the transmission of data and so on. But obviously it can potentially resolve some very important questions around epilepsy, particularly relating to the timing and pattern of seizure activity. It's been very difficult to answer these to date because really the frequency of events is relatively low. A routine EEG done over 30 minutes on average infrequently captures seizure activity because the bursts of activity are relatively infrequent, and clinical seizure activity is even less frequent, separated by days, weeks, months, or even years. So capturing events is very hard. Doing this through scalp recordings is difficult because of the low fidelity of recordings and their susceptibility to artefacts, so this is a big challenge. As well, we're often uncertain about the relationship of the EEG changes to the clinical state of the patient, and determining this from the recordings without video confirmation or some other form of confirmation is tricky. But if we're able to do it, then potentially we could understand the dynamics, understanding epilepsy, understand the biology of the condition a bit better, and perhaps even provide better therapies. Now, the periodic nature of epilepsy is very well understood, and there are some really interesting pieces of very old literature dating back to the 1700s even about these striking patterns of the periodicity, most well known to clinicians through catamenial epilepsy, where there's a strong relationship to menses. As you'll see, that's in fact not unique to women, and in fact it's quite frequently seen in men as well. For that reason, this man in the early 1970s commented that although computers hadn't been yet used to study the patterns of epilepsy, it leaves a good many male epileptics with 28-day periodicity staring at the moon waiting for an answer. But there was some very interesting work done in the 1920s by Brain and Langdon Down, looking in a institutionalised colony of patients with epilepsy, as was the fashion at the times, looking at these records taken from patients through direct observation, looking at the patterns of circadian rhythms in the seizure activity of the residents, and they saw these striking patterns, which really, looking at our data now, we didn't do much better than it really, and it's incredible the insights that were received through very careful clinical observation over 100 years ago. More recently, some very interesting work was done in the early 70s by Jan Stevens and colleagues in the States, where they had what must at the time have been very sophisticated data for the continuous transmission of EEG data using FM transmissions done in patients with primarily generalised epilepsies in a ward setting, but they also observed these very striking patterns of seizure activity. But we're not able to take it much further than that. I think there's great importance and interest in the area because we understand that biological rhythms are important to lots of diseases, and that if we can understand the rhythms and timing of the problem, then perhaps we can apply very specific and focused therapies. An interesting table from this article on chronobiology and chronotherapeutics outlines the close relationship of many diseases to circadian rhythms, but interestingly it doesn't feature epilepsy amongst them, which I found rather striking, as obviously epilepsy and its relationship to sleep is an extremely important factor. So with colleagues Sam Berkovic, Terry O'Brien at Austin and Royal Melbourne Hospitals respectively, now 6 years ago we started a study of an implantable device to predict seizures. And with this system, you can see here that there's a unit like a pacemaker that's placed under the clavicle, a handheld unit that the patient has that records the data transmitted from the implanted unit, and a set of electrodes which are placed subdurally. So we had to do a craniotomy. You can see the craniotomy here. We've done a craniotomy and electrodes are distributed over the surface of the brain, you can see in these images. So quite invasive procedure. And the fact that people were prepared to undergo this procedure to have seizures predicted gives you some indication of how much impact the unpredictability of the condition has on patients. And we recorded very high quality data from these patients continuously over periods of up to 3 years. And so this is quite unique. Ambulatory EEG data of this type had never been recorded before. Normally we do acute intracranial recordings over a period of maybe 7 days, exceptionally 10 days. So this is quite unique information. And we learned lots of interesting things. This is a table looking at the frequency of seizures as stated by the patients prior to entry into the study, and then what we recorded. And you can see that patients were recording prior to the surgery, prior to the implantation, 7 seizures a month on average. During the study in the first phase, reporting no seizures, but we were recording 126 from the patient. The handheld unit had an audio recorder so that it was activated by detected events rather than predicted events, and so we were able to listen to what was going on around the patient at the time of electroencephalographic events. And surprisingly, through this, we're able to recognise lots of clinically unrecognised seizures. We could hear the patient falling, grunting, convulsing. We could hear other people coming to help them. So that was quite startling that this small additional piece of information gave such dramatic insights into the clinical scenario. As well, though, some patients were reporting seizures and not actually having any when we put them in. So this was another striking feature, and this is well shown in this graph here, demonstrating the reported seizures down on the x-axis here versus seizures that were recorded with a device that we were able to clinically confirm on the y-axis. So you can see this is probably still an underestimate, but you can see there's an order of magnitude difference between them, and the correlation between them is quite poor and in fact varied month to month and between patients. So it's quite startling how poor the correlation is, given that at the moment all of our therapies and new drug trials are based entirely on patient diaries. As far as the prediction went, the prediction was successful in most patients, extremely successful in some. There were some patients where we weren't able to generate successful algorithms for prediction, but there shows the average seizure prediction from the patients, and you can see we've got about 90 minutes warning of seizures, so it's quite long prediction, not seconds or a few minutes. It ranged between 8 minutes and 4 hours, so very useful prediction horizons for the patient who potentially could take an acute agent during that phase interval. As well, though, we learnt lots of other interesting things, and first of all, we learnt that the EEG wasn't stable after you put in electrodes, and that mightn't come as a surprise, but it was a surprise how long it lasted for. So it lasted for 60, 70 days, out to 100 days for some patients. This was seen as well in canine studies, though we weren't aware of that at the time. So the fact that the EEG was unstable, shown here, this is just a graph of the total power of the EEG, just to show you a nice demonstration of how it normalised over about 100 days in this individual. But it meant that unexpectedly we weren't able to use data acquired in the initial phases of the study for prediction, which almost causes us to abandon the study. Fortunately, things levelled out. But we were able to see these other striking patterns, and you can see shown on that graph the different colours of those dots represent seizures of different clinical types, so we were able to categorise clinical, subclinical events, shown there in blue and red respectively. And you can see these striking patterns, so there's the distribution of events over a year, and if we take a month of that we can see a similar pattern. Similarly, if we break it down to a fortnight, and down to a week, we can see these recurring patterns, so there's a very fractal structure to the distribution of the seizures over time. And that keeps going down if we start including spikes and so on. So these very interesting patterns, which on the surface appear random, but which can be shown to have a deep structure to them. As well, there are these striking features relating to time of day, and you can see in the grade bar there the seizures that were occurring during daylight hours in this one individual, most of the events during the daytime hours and some during sleep, whereas spike activity in this individual was predominantly during sleep. So we were able to also clarify the relationships between spikes and seizures, but found, as you'll see, that this is incredibly varied and different between patients, so there's very individual patterns. Bursts of activity. You know that I've been interested in the relationship between seizure activity and its correlation with earthquake activity, because we see similar patterns of activity both in the interval between events and the magnitude of events, which can be characterised by a relationship such as a power law relationship. This is showing a power law relationship for earthquakes. So power laws describe essentially that very small events are extremely frequent and very large events are very rare, and they're related by this logarithmic plot. And finding this relationship is important as it has several implications. One of them is that it tells you that the dynamic system underlying it might feature self-organised criticality, but it also might help you in prediction. We see this in lots of natural systems, and I'll show you another interesting example of that shortly. Here is the data plotted out for the 15 individuals in the NeuroVista study, showing the frequency of events and the intervals between them, again plotted out in the smaller blue graphs on a log-log plot, showing this same strong power law relationship between the frequency and interval of the events. And finding a power law relationship is useful as well, because it might contain within it a Hurst exponent, and these mathematical features relate to underlying rhythms that display a long-term memory in the system, so that you can see that past events are influencing future events. And you can extract these from data. This is an example here you can perhaps appreciate in this artificial data here. Actually, that's not artificial data, that's the river data, that's the Nile, the level of the Nile, and the Nile exhibits this feature where you can see there's rhythms within it, and you can extract from it a Hurst exponent, which is done here using a wavelet function to demonstrate. We can see through the slope on this line that there's a strong Hurst exponent. So these Hurst exponents tell you that these deep memories involved in the system. This is just white noise, and there's no slope to this line, so the Hurst exponent is not present as would be expected. Here you can see in the patients with epilepsy, not all of them, here's a patient who doesn't have a Hurst exponent, but many of the others had Hurst exponents, telling you that there is long-term memory in the system and that past events are influencing future events. And this has been best described by the mathematician Mandelbrot as the Noah and the Joseph event, where long intervals lead to other long intervals. So if there's a drought, there's likely to be another drought, and if there's floods, there's likely to be more floods, as we see with El Nino and related weather effects. For these patients, these are the sort of cycles that the Hurst exponents were present over, and you can see they range from hours to days. You can see down in the bottom here these very striking rhythms which underlie the patterns of seizures. There are other patterns, so we were able to look here again at individual patients. These are the 15 patients with the duration of their seizures plotted against the time of the study. This is plotted logarithmically, and the important thing is you can see that their seizures fall into several orbits. So there are some patients who had a single orbit of seizure activity. Others had a couple of clear different patterns. Others were a bit of a mess. And some probably contained 3 orbits in there. So there were lots of cycles, and you can see that they're relatively stable for the patients throughout the duration of the study, and as well that they fall into fairly well-defined orbits. So that was quite surprising, and most interesting about it was that the patients who we obtained really excellent prediction in, 100% prediction in a couple of the patients, were those who had these very uniform populations of seizures. So these were the most easy to predict, and probably relates to the fact that we've got several different patterns of seizures running simultaneously, and the algorithms need to be generated to cover each individual pattern. You can model these and show that these are genuinely different categories of seizures, and we'll come back to why that's interesting and important later on. And you can also look at the relationship between clinical events and duration, and of course it would be easy to presume that, and logical to presume that longer events are more often clinically manifest, but this is a rank order plot of the duration of seizures, so all the seizures plotted out longest to shortest on a logarithmic plot there. And the red events are clinical events and the green events are subclinical events. So you can see these are 2 separate patients. You can see for the first patient, all the clinical events fall in this longer population of seizures. But for this patient, both the very shortest event that you might see down there as a red cross and the very longest event were both clinically identified by the patient as relevant. So it's not standard, the patients are all different, and there are these very striking patterns and disparities. Similarly, the interseizure intervals show these same sort of patterns, again on a logarithmic plot there of the interseizure interval on the y-axis against duration of the study on the x-axis. And you can see this very striking palisading of the clusters of seizures. It's possible to look at the relationship between this. One question that had always interested me was whether it's true that patients often observe if they have a long interval between seizures that the seizure they have then will be more severe than ones that are close together. And in fact that is true for some. You can see this patient here in this quadrant, all the clinically significant events were also those in the longest duration phase. True most of the time for this patient, but for other patients the relationship didn't hold. So again, very, very individual patterns. It's interesting, here's another natural system that shows a similar sort of pattern, and as I mentioned, these systems are thought to, or these patterns are felt to relate to self-organised criticality in the system. So these are the eruptions of Old Faithful with interval versus duration. You can see a very similar pattern as to with the seizure activity. So it's just an interesting aside. Here for the patients are plotted out for individual patients. The grey bars represent day-night. They're 24-hour plots on the x-axis, and the seizures every day of the recording going up the y-axis. And the important thing here to see is that throughout the entire duration of the study for these patients, they kept a very stable pattern of seizures. And it's interesting, the patients rarely observe this, that there are certain hours of the day that they would never have seizures, not throughout the entire study. So very interesting, and others were less well defined, and seizures could occur at any time. But obviously having this information enables much more powerful predictive measures to be made. It's the relationship between spikes here shown in black and seizure activity shown in blue, again on a 24-hour time scale. You can see, again, very individual. Some patients had a very close relationship with synchronisation between increased spiked activity and seizures, whereas others, they were quite out of phase. So how does this help? Well, it might help us understand a little bit better what's driving these seizures. So here's some illustrations of the short and long events. And I've shown you how we can take this information out and model it, and they form very distinct categories. And we can use then neural mass models to understand this activity better, and using real data and these neural mass models understand through these very simple models of cortex what might be the relevant components contributing to the seizure activity and the cessation of the seizure activity. So we can, through the analysis of this data, we can attribute various weights to the elements of the model and display this graphically as here. Here showing the difference between long seizures and short seizures in input to the pyramidal system, and an inhibitory input to the pyramidal system shown on this side. And you can see in these heat maps an illustration of the connection strength, the red line illustrating the onset of the seizure and the next red line illustrating the offset of the seizure. You can see these very different patterns between the long seizures and the short seizures, so the dynamics of the seizures are quite different. And this is a difference that evolves after the seizure has started. It's a difference that's there immediately, maybe even prior to the seizure starting. There's another illustration of that same information, looking at the various components of input to the model from the various elements, both related to the onset and the offset of the seizures. There's very distinct patterns of activity relating to the onset and offset of the events. And if we look at that with these unimodal seizures, so I showed those patients who had a unimodal population of seizures, and we look at the various components, you can see this very striking pattern of activity here again. And remember, these are patients who we obtained very good seizure prediction in. Very striking and constant patterns, but if we look at a patient with bimodal activity, again we can see these two very distinct populations, and you can appreciate even from these heat maps here, you can see that there's very, very different patterns of involvement of input to the system, both prior and during the event. So they're very different events, but that difference is apparent from the outset or prior. So this has been a very interesting and perhaps useful way to analyse it, because obviously if we can understand how seizures stop, we might be able to develop better therapies too. Here looking again in that patient just at the onset and offset of the events, these differences in the patterns. So this is all very well, but how will we do this? I mean, we did 15 patients. The device was very expensive. The company collapsed because venture capitalists and other investors didn't see that patients would be willing to have these devices inserted. Obviously they were, and they would be, but the invasiveness of the procedure was a no-go zone, unfortunately. So we've been working on a less invasive system. We've been working on this for a while, but it's not been possible to reveal too much of it to now through the usual commercial reasons. But this is based around a bionic ear device. You can see the bionic ear device here. You can see this is just a mock-up with some different kinds of electrodes over the skull, but you get the idea that we can insert this under the skin of the scalp and deploy the electrodes under the scalp and record seizures chronically. And I hope you can see that just recording the seizures chronically is valuable, that we can learn a lot through them, even if we don't have a lot of discriminatory information about their lateralisation. Simply knowing when they occur and their duration gives very powerful information that might transfer very directly to patient management and potentially also to specific therapies later on. We deploy the electrodes under the scalp as well. I can't show you pictures of our finally manufactured model, but we are manufacturing currently these devices, and that's our final device illustrated there. So these electrodes are tunnelled under the scalp, you can see in this illustration here. So we've developed tools that will deploy these electrodes very quickly under the scalp, and the basic bionic ear system is used to collect and wirelessly transmit signal to an external unit, which at the moment is a tablet PC, but later on will be a phone. And it will hopefully allow this sort of analysis. Whether it will allow prediction yet, we don't know, but we suspect based on the information we have from the NeuroVista study that will be feasible also. And we've been studying these in sheep for a while, so it's a bit of a tangle in the sheep's head. Clive May, of course, has been incredibly helpful here at the Florey in helping us get there, and I've got to put in a plug now for just how critical it is to have large animal facilities for doing these studies on site. Absolutely critical. We could not have got to this point without the availability of these systems. So at the moment we've studied these, so we've had these in sheep for over a year, and as I mentioned, with Cochlear are manufacturing the device for us at the moment. So we're well underway with that and hoping to get some clinical trials started with it next year. So, the invasive ambulatory monitoring is incredibly valuable. Quite apart from the possibility of predicting seizures, you can learn an enormous amount of useful information from simply recording accurately when seizures are occurring and their duration. And this provides a lot of unexpected information relating to the biology and the dynamics of seizure activity. It might open new therapeutic opportunities eventually, because at the moment our therapies are based on chronic therapy for an unpredictable event, providing uniformly sedating and sometimes personality changing therapy. If we could provide something intermittently on demand, it would be so much better, obviously. These systems could be linked with peripheral devices. I mentioned that, you know, if you can include a bit of audio, you can learn an enormous amount, and if you could put some accelerometry or other sophisticated environmental information into the system, whether that be through a watch or a phone, you'll make these much more powerful units. As well, we have less invasive devices on the near horizon now. Of course, there's an enormous number of people involved in studies like this, but I'll just embarrass some of the... One problem working with engineers is they're all guys almost. So I'll mention Philippa there in the middle, and Pip's a PhD student working on this at the moment, and she's been responsible for a lot of the work that we've done around the analysis of the relationship between spikes and seizures. Of course, Ewan's been involved in the burst activity. Andre's a mathematician who's been very involved in the project now for some time. Farad, I didn't realise was going to be there, so sorry Farad, I would have got a better picture of you. Farad is another engineer. Tony Burkett, you'll know from engineering. Alan, who's been involved in these projects from the start, another engineer who's really our project manager under the implantable device system. David, I've already spoken about. Chris Williams, you might know through his involvement in the Bionic Eye project. And Dean Freestone, who of course has been so important with our modelling studies. Ray Boston's a biomathematician from Melbourne originally, but who's been in Pennsylvania for a very long time, but who comes back and works with us regularly from the States, and he's been a genius helping us get all of this together, and particularly understanding the statistics of it all. So thanks once again for the invitation, and I guess I could take some questions.
Yeah, thanks, Mark. I was wondering about the revisiting of all of this and in terms of innate versus being secondary behaviour. So, are you able to correlate it with endogenous rhythms like cortisol and overlay with behaviours including stress, but also glucose before and after feeding and sensory cognitive physical activity? Are you able to identify the triggers?
Well, the short answer is no, but the cortisol one, I guess, is the most important. It's difficult to think what biological rhythms run on 28, 40-day cycles, and they seem the most prominent ones. And I might add, although it's very different data acquired through the NeuroPace study, they only have time-stamped data and looped for seizure at most information. So they've got a very limited data set, but they've seen very similar patterns. And it's very interesting to know what could underlie that. Similar patterns I've seen in idiopathic generalised epilepsies as well, so it's not just a phenomenon of focal epilepsies. The cortisol is the most obvious target, and with collaborators in Exeter, in fact, we have a device which sits peripherally and samples through skin cortisol, and next year we're going to start studying exactly that, seeing if there is a relationship to cortisol. There's a lot of other work showing cortisol is probably related to excitability, so it's target number one.
So presumably if you took this environment approach and gather all this environmental data as you go, you actually be able to use machine learning to work out what specific triggers work.
Yeah, I think that's right.
I thought it was very interesting that you have some patients that report that they're having a seizure, but you weren't actually.
Yeah, I guess I think Sam's here. He'll confirm that this is actually a common problem for us. People reporting events where you get them in hospital and their events sometimes are not organic at all, and sometimes a migraine, or sometimes other things they're misinterpreting as seizures. So it's a surprisingly common event. It's a bit, I mean, that patient was in fact having some seizures during the thing, but you do have it where you bring patients in and they've sometimes been treated for epilepsy for years or decades, and you find that none of them are actually seizures.
Very interesting data. By analogy with the visual system, where the optic nerve doesn't carry raw information from the code itself, but locally pre-processes, could you introduce a pre-processing step right back into the skeletal, which would narrow the events of the brain capture, but presumably with less noise and
Do you mean could we make the electrodes so that we could do that better?
we could put a local device immediately beyond the electrodes, which process.
Oh, yeah. Well, how much processing you put on board the device, I guess, is simply a function of time at the moment, isn't it? So, I mean, increasing amounts can be put on board the cochlear system. There are cochlear systems which they've experimented with for other reasons, which have accelerometry on, for instance, and can analyse it on board. The problem is at the moment phones have so much on them that if you can link it to the phone, far more sophisticated technology can be put on board that, and everyone carries their phone anyway. But it's an interesting idea that you could contain all of that in the implant and have SMS and GPS capacity so that people could be notified, for instance, if someone had collapsed with a seizure.
All right, one last question. My last question. How invasive is that procedure? I should imagine it's quite invasive, putting electrodes subcutaneously right through the scalp.
The scalp? Well, look, we've only done it in sheep, obviously, so far. Our surgical colleagues tell us it's no trouble at all. We're about to start some cadaveric studies with it, but they won't complain much, I guess. Yet to be determined. I think, it's an unfortunate feature of the commercial world that unless this is a very short procedure that doesn't require overnight hospitalisation, the economics of it all suddenly get out of whack and it becomes difficult to mount a case for it. So we're very, very interested to make sure that it's a straightforward procedure. I think it will be. I mean, as I say, we have a special tool. You make a cut and you feed through the electrodes in one swipe, essentially. So should be easy. Famous last words.
Okay, so it's a real pleasure to introduce our final speaker, Professor Read Montague. For any of you that have spoken to Read over the last few days, you no doubt noticed his remarkable enthusiasm and passion for research. So after the other night, he mentioned he was particularly enthusiastic about the research, his more recent research that he'll be presenting today. So ever since then, I've been extremely, very much looking forward to hearing about what he's got to say. Now, as with all the preceding speakers, Read has a CV that's jam-packed full of large grants and high-impact papers, including multiple Nature and Science papers. But to me, the standout feature of Read's work is the frequency which his name appears in in seminal papers across a really diverse range of fields. So really a number of important papers in the cognitive neurosciences, but also in psychiatry and computational neuroscience. His early training includes a background in mathematics and then biophysics and neurobiology, and this diversity has really continued throughout his career and is reflected in his multiple official academic roles, where he currently quite remarkably holds a prestigious, one of that's not the remarkable bit, the prestigious bit, but that he has a Wellcome Trust Principal Research Fellowship at the University of College London that sees him spend 1 week every month in London, and he's been doing that since 2011. Now, his primary base is in Virginia Tech, where he's the inaugural holder of the Virginia Tech Mountcastle Research Professorship. He's the director of the Human Neuroimaging Laboratory at Virginia Tech and the director of the Computational Psychiatry Unit. He's also a professor at the Department of Physics and a professor of psychiatry and behavioural medicine, to give you a sense of his breadth of interests and research. Now, beyond his research achievements, I also just wanted to thank him for his engagement and contribution to a number of different mentoring events over the last few days with our early and mid-career researchers, so I'm sure he's helped to inspire some of our students. And I just want to thank him for that and also provide him a warm welcome to Melbourne. And if you come up here, I look forward very much to hearing your talk.
Can you guys hear me? Yep. Is there a microphone to pick me up here? Do I need to? Yep. I've lost the AV guy. Sorry. All right, let me just. The appearance of my name on those papers, high profile or not, is a testimony to my ability to choose science friends. That's what it is. Okay, so this is my. Today is Thursday, right? So this is my 7th official presentation since I arrived on either Monday or Tuesday when I arrived. I mean, I started with enthusiasm, but I'm feeling it now. I'm actually quite enthusiastic to talk about this. This is not quite as dramatic. I'd like to say, is Mu-ming Poo still here? Yeah. So I don't know if you remember me, but I'm a consumer of your work for my whole career, and I feel quite honored to be a bookend to your opening the door to the symposium. So thanks to the organizers for letting me be at least on the lineup after Mu-ming Poo. I'm also going to talk about learning, and I'm going to also talk about prediction, and but at a different time scale. So what you saw in the first talk is you saw a lot of work on correlational learning rules and the physical events in the brain that are changing synaptic connections based on correlations, and then you saw something that looked like a shift in that, a little prediction rule. You saw a little kernel that he showed you with what is now called short-term dependent plasticity, which is not symmetric in time, which gives it the possibility for doing prediction, as you heard Professor Poo. Did we say Professor Poo? Okay. I'm going to talk about dopamine. And I'm going to start by reviewing, at least relatively rapidly, theoretical work that I did with Peter Dayan something like 23 years ago. And I'll step through sort of what we made claims about. And then we branched off. He kept going one direction, I went another. And we've sort of come back together again over the neuromodulator serotonin. So I'm going to focus on dopamine and serotonin. This is going to be a rather narrow talk. I'm going to be looking at conscious human beings making choices while we record, using some new methodology, serotonin and dopamine fluctuations in the caudate. These are patients that have either essential tremors or Parkinson's disease, and they're being affixed with deep brain stimulating electrodes. I'll get back to that, but I just want to say that's what the whole thing is talking about, but it's talking about a principle. So one of the things that I worried over about correlational learning rules, Hebbian learning rules that you heard talked about in the first talk, was that in many instances it's a tautology, right? So I'm going to look out in the world and in things that tend to correlate with one another consistently and invariantly across epochs of time sufficient for me to call it a correlation. I'm going to wood burn those into my head. And it always bothered me, that idea, even though I admire Hebb's approach, that if something was occurring and correlated that much, why bother storing it, right? Why bother storing things that you sort of already know about, that the world will deliver to you? Well, there are answers to that. I'm not going to give those now. One is generalization. You want to generalize over things. But I got very interested in prediction, and so I made the claim with Peter Dayan back some 20-some-odd years ago that we started fitting models to diffuse neuromodulatory system. That is, systems that sit in your brainstem and project throughout the cortical mantle into your striatum and deliver neurochemicals like dopamine and serotonin. We assumed that they were a broadcast system that was sort of one-to-many broadcast, sort of a scalar signal, and we wondered what they would be doing, and we early on decided they were doing prediction and handing out a prediction signal to the rest of the brain. This is before we had any data. Okay, I won't go why we thought that. Well, that idea fits the firing rate changes in dopamine neurons now in a bunch of rodent models, but initially in primate models. And the idea is outlined here. This is the computational background. So I'm going to show you the computational background and I'm going to show you bold imaging experiments, functional MRI experiments, then I'm going to show you how those target where we put our electrodes. The idea was that activity in these neurons, this is dopaminergic neurons, pause, burst activity, and no change activity represented a continuous emission of information. Differences in expectations about what was going to happen, positive, negative, and no change at all. That is, there were prediction errors over reward. Okay? It's a simple system. The idea was it was a simple system. The cortex would frame a problem, send this information down to brainstem nuclei, it would parse it up and be emitting errors, and the rest of the brain has to be able to know what problem, in essence, was on the active cue. All right? The new idea there, aside from relating it to something called dynamic programming, which we'll come back to, one of the new ideas there physiologically was that these neurons are emitting information all the time, even when they don't modulate their activity. So no change at all in their activity to downstream structures is a signal that the downstream structures can interpret as things haven't changed. Okay? Here's Hebb's idea. Hebb's big step. This was at the end of his career. Actually, this is published a year after a book by Jerzy Konorski, who was Ivan Pavlov's star student and who has disappeared into the fog of history. I don't know why. Konorski published the year before this, in 1948, a book where he published basically the differential version of the Hebb rule. Hebb's, I think, insight here was to take the psychological findings as expressed in operationalized experimental psychology experiments and turn them into a physiological hypothesis. That was a real step that he made, and it took an insight on his part. Konorski's book wasn't quite so clear in that regard. So we got interested in this system and started writing papers saying that we think that these diffuse neuromodulatory systems are producing a particular kind of prediction error signal. Not just the difference between what you expect and what you get, but a difference between what you expect now, what you expect as a kind of model into the near-term future, plus what you didn't account for. So it's a successive prediction model. You can relate this to lots of methods in optimal control that have been developed starting in the 1940s, actually, coming to kind of a fruition at the end of the 1950s, and then being expressed in various kinds of communities that do optimal control problems. And we wrote papers like this, and I have been to mentoring sessions. So I installed 3 slides with mentoring issues in them. Issue number 1, using aperiodic reinforcement for directed self-organization during development. Not a pithy title. Do not write papers that have titles like that. All right? All right, but that idea, this is in 1993, is we present a local learning rule that takes Hebbian learning and augments it. It takes a Hebbian learning rule and it puts another factor on it that's a prediction error signal that carries a sign. So you can take Hebbian learning, correlational, and you can multiply it by a factor that changes that can modify the direction of learning. We saw now, though, that in glutamate synapses, you probably can do local learning because of things like short-term dependent plasticity, which was completely unheard of at the time. We then wrote papers on that. This is a paper with Terry Sinofsky. I wanted to show you this paper because of another lesson. See this lesson here? Volume 1, number 1, page 1 in the journal Learning and Memory. Don't do that either. Nobody reads volume 1 of any journal. Volume 1 of every journal is struggling to be midwifed into, progress. We have a journal now, Computational Psychiatry, and volume 1 is struggling to be born, believe me. Come to the MIT Press booth where I'm going to sit and cheerlead for the thing in San Diego this year. All right. But what we did in this paper, what we did was we took these ideas from 91 and 92 and 93, and we made the suspicion that the whole brain was doing predictive learning. There's now kind of an industry, part of it coming out of Queen Square from an office down the hall from me of Carl Friston, of a Bayesian brain hypothesis where the whole brain is thought to be doing prediction at multiple scales, and we conjectured in here that what that might look like and the kinds of physical events that might mediate it. Then you can take, and this becomes a connection to behavior here, you can take those same ideas and go right into other organisms that have neurons that are neuromodulatory. The bees have neurons that deliver octopamine. There's an subesophageal ganglion that deliver octopamine to the whole bee brain, which isn't gigantic. And they mediate learning about odors, learning about visual patterns, but mainly learning about nectar. Where is the nectar source? And they look like they do prediction error emission, and that controls through the release or non-release or diminished release of octopamine. It also controls learning in the mushroom body of the bee to control what they learn on and where they'll adjust and adapt their behavior. And this is the next lesson. So this is a paper showing the next year and a model of primate dopamine neurons that, in fact, you can account for 25 years of primate neuron activity as a prediction error signal that I've been sketching for you here. This paper took 6 years to get published, and we wrote it 11 times. All right. In fact, the reason, this is a mentoring session, the reason that this paper, I was so confident in this paper here about bees was I knew that I had this model sitting in the background that I was fighting with, people who probably to this day hate me, on the primate paper. So I knew it really accounted for a lot of data in a primate paper. I was very excited about it and disappointed that I couldn't get that published. So I was sort of naive. So if there's any lesson here, it is keep going and pick better titles for your papers. And then we paired up in a review paper with Wolfram Schultz, who is the pioneer here, the physiology pioneer for sure. And I'll just say one thing about that, why I think he is the physiology pioneer. Are we sharing this glass of water here? So, we are now. In the early 1980s, there were lots of primate physiology labs, Crutcher Alexander and Malin DeLong's group in Emory. I forget where Schultz was at the time, Anastopoulos Georgopoulos, who's now in Minnesota. And they were looking to record from dopamine neurons in primates as a model of Parkinson's disease, and they sought correlations with metrics of movement. Parkinson's disease, you lose dopamine neurons, you can't move so well. Surely there'll be a correlation between dopamine neuron activity and the metrics of movement. And everybody died on that hill. There's nothing systematic there, except for Schultz and Ranulfo Romo. And so they suspected something. It was something to do when they were shaping the animal and whatnot. And so it's a testimony to them hanging on to it that got clear. So here's the picture. Here's the physical picture here. We have dopamine neurons that are emitting and modulations in their spike rate, prediction errors and future reward. Let's call it near-term future reward, from now into 30 seconds into the future. Okay? These run out to the terminal. They release dopamine, and now you have a fluctuating signal here. I've shown it in orange. And then any learning rule that adapts according to, in this case, pre- and postsynaptic correlation plus the fluctuation in dopamine and adjust a weight accordingly will learn a prediction. Okay? Once you learn a prediction, then you're learning a parameter that tells you where to go into the near-term future. So instead of learning correlations, which are static and you have to add something to that to make it learn temporal sequencing, you learn and store predictions. Any tripartite rule will store stuff. So you can imagine, I'm focusing on synapses here, but you might focus on extracellular matrix molecules. There are lots of ways you can modify extracellular matrix molecules and scaffolds that look like a tripartite rule that might well depend on neuromodulators at dopamine and serotonin. All right. 2 more slides on the model. I think it's important that I go through and just walk through that this all took place as a bunch of models. It wasn't that well supported at first, but has become so in the intervening time. The way the model is conceived is something I really liked. First of all, it's mathematically explicit. I'll show you that in just a second. But it says that an animal or an organism has states available to it, and that the value of any state is, in a history-independent way, dependent only on the future of that state. So I don't care how I got into a particular I'm walking out of the microphone. I don't care how I got into a particular state. I just care the value of things that I can reach into the future from that state forward. Okay? And we're going to take the expected value or average value of the future discounted states. They're going to be discounted so that things in the near-term future are more valuable than the far-term future. And I've written that here in words. When you write that as an equation, you say the value of the state at S sub t is equal to the expected value of the rewards that you experience at times t, at t plus 1, at t plus 2, at t plus 3. Okay, it's written discretely. Gamma here is a discount factor between 0 and 1, so it just says things coming a little later, more distally into the future, are less valuable than things now. Okay? We know that's true behaviorally. Okay? That's it. That's the assumption. That's the explicit goal of learning, is to estimate the value of states. Once you know the value of states to you, you know how to trade off state 1 versus state 2. You know how to trade off whether I make that transition or I wait there or whatever. That equation enjoys a recursion relation that I've drawn right there. Okay? If the first one's true, the second one has to be true, and that's called the Bellman equation. All right? That is a representation, in a sense, of how you do optimal control if you add some more things into it. Okay? This is Richard Bellman, circa 1957. Okay, so if the left-hand side is equal to the right-hand side, then on average, in a real system, if you're making an estimate of that, if you just subtracted them, then that thing ought to be sort of 0, and I put quotes around it for the obvious reason. That is what we think is coming out of the spike modulation of the dopamine neurons. Okay? That's a pretty explicit model. It allows you to construct behavioral scenarios and gambling games and decision games and whatnot, and then model them expressly and go in and ask, do these neurons emit control signals like this? And fortunately, or unfortunately, this is a really important system. If you lose your dopamine neurons, and you keep losing them, life isn't worth living. You act as though life isn't worth living. If you lesion them in an animal, the animal acts as though nothing has any rewarding consequences to it. These are crucial neurons for your survival. All right? We had the opportunity some years ago, first in Houston, and then after I moved from Houston to Virginia, to take a method called cyclic voltammetry from the rodents and build an electrode that would go into a human brain. It means a lot of things. We have to make it longer. We have to do a bunch of stuff. This is with my collaborator, Paul Phillips, from the University of Washington. We did that. We developed it. And we went right into human beings being implanted with deep brain stimulating electrodes for the treatment of Parkinson's disease or for the treatment of essential tremors. Parkinson's disease is a dopamine disease. If you have Parkinson's disease, you're losing dopamine neurons or different versions of Parkinson's disease, but that's still true. And the main treatment, by and large, is dopamine replacement therapy. Not so with essential tremors. Essential tremors have also a kind of a panoply of expression, but it's not treated generally with dopamine replacement. Okay, so it's thought that essential tremors, sometimes essential tremors will develop into Parkinson's. So it's a murky relationship there. Virtually nothing is known, as I've written here, virtually nothing is known about which part of the computations from the animal literature that we think dopamine neurons are capable of supporting are missing, perturbed, intact, sustaining, different across different disease states in Parkinson's patients, making this an opportunity. I will say one thing about the patients. These are patients typically, but not uniformly, these are patients typically whose symptoms are no longer relieved by taking dopamine precursor molecules. So the drugs aren't working anymore on them. Okay, they're going to play a little game. They sit in the surgical chair and they're going to play the following game. This is a game I played in people, and almost 850 people now doing functional MRI experiments. So I have activations that track control signals in it, and I know where these electrodes would be most likely to pick up a signal that would be useful to me. Here it is. You have $100 at the beginning of the game. You invest some fraction of it. Here this guy's gone to like 45%. A market fluctuates up, in which case you win, or it fluctuates down, in which case you lose. That's it, rinse or repeat. Okay? So it's a relative, sort of a half-market task. If you think the market's going to go terribly, your option is to cash out. You can't short what economists would call, traders would call short the market. There's no shorting the market. You just express your confidence in what's going to happen, with zero being the way you get out. And you can sit there at zero the entire experiment and keep your $100. Okay? What it allows me to do is when you have a gain, we have gains here, you have another quantity on your fixed budget that I'm calling a fictive error, and that's what you didn't get but could have on a gain had you bet higher. All right? Now what you can immediately start guessing is people are different and their relative sensitivity to the things they leave on the table versus the gain that they got. Some people are really run toward, they hate leaving things on the table, and some people really like big gains, and these things trade off for one another, and so it's a distribution of sensibilities there. we call that the fictive error. What you're going to see is it ended up being a pretty good guess as a structure of this game. So I want to put in your head the following. What we think is that dopamine neurons are emitting a reward prediction error. So if I'm playing this game, I set the bar, I get a gain, I set the bar, I get a loss, I set the bar, I get a loss. I can look at my average return and look at my expected return, and I can look at fluctuations in the outcome around that. And that's a natural proxy for this prediction error that I've been showing you. So that's what I'm calling reward prediction error, RPE. But then we have this counterfactual piece, and I'm going to subtract that, okay? In other words, this is the difference between what I could have gotten had I bet all in and what I did get because I bet, in this case, 42%. And that difference is my counterfactual error. In a sense, it's my foregone gain. And so I'm going to reduce my reward prediction error by the amount that has been foregone. If I bet all in, then that foregone gain is 0, and I'm just looking at a pure reward prediction error that becomes relevant later. All right, so let me just show you a couple of behavioral results. Behavioral result number 1, that fictive gain piece, because I told you some people are sensitive to that. What does it do to people's behavior? So if I look at people's behavior and just do a linear regression on it, trying to predict how they're going to adjust their bet on their next move, what controls that? And the answer is 2 things. One is the market, just how well are things doing on average. If the market's really high and you're making money, you're happier. That regresses onto the change in your next bet. And the second thing is the fictive error. And I've divided it up here into low, medium, and high fictive error. And these are the regression coefficients normalized on the y-axis. Okay? That's just on gains. I'm just focusing on gains right there. If you were to go look for that signal, these signals, in what's now called model-based functional MRI, I'm not sure why it's called that, actually. It's just regression. Here's the kind of thing you would do. So what I've drawn on the top here in market changes is the relative change in price. Actually, I've got to put my glasses on, sorry. It's that foggy. The relative change in price at time t, right? The relative fractional change in price. I've regularized the times here. The times it's a free response experiment. You hit whenever you want, but I'm just showing you where it is. So that's positive, that one was positive, that one went down. Here's the bet on for each of those things. This is for an individual subject. Now I'm just showing you computed the fictive error. And here's the functional MRI trick. I now take the canonical hemodynamic response function. I convolve it with each of these functions here. I produce predicted hemodynamic response, in this case with the fictive error, and I go fishing for that in every voxel of the brain. So this is a very different experiment than like a 2x2 cognitive design where I'd say I'm in state X and I'm in condition 1 or condition 2, and now I'm in state Y and I'm in condition 1 and condition 2. Instead, these are parametric regressors that are defined by the elements of this model that I just showed you, and this is fit individually to each person's sequence of choices and experienced gains and losses. All right, there's a big difference there, but when you do that and you go looking for things, let me just draw your attention to the bottom here. This would be a signal that you would call the reward prediction error, and this would be the signal that we would call expressly the fictive error. These things are collinear sometimes, and so I can separate them from one another. What I'm showing you here is the entire region here, so some of the fictive error is in this big blob here. You can see it's in the striatum. The fictive error ends up restricted to the ventral part of the head of the caudate nucleus. This is something I did in 70 people. This is in 2007 paper. And so when we had this opportunity to go into the Parkinson's patients, there was a lot of talk about the surgical trajectories, and one of them goes right by the head of the caudate nucleus. So they're heading to what's called the subthalamic nucleus to put this deep brain stimulating electrode in, and I was going to go down in their guide cannula. We weren't going to poke any new holes, but we're going down in their guide cannula, and we get about 15 minutes. We poke about half a centimeter out the end. The electrode is here. This is a human hair on the left. This is the fused silica flexible electrode, and here's the business in here. This is anywhere from, in our hands, 100 to 150, 100 to 120 microns in length and 7 microns in diameter. It's a carbon fiber. We're going to cycle a voltage on that. I'll come to that in just a second. This shows you the surgical trajectories. You draw your attention to the coronal section here. So these green things, which which are a bit faint here, are the pre-planned surgical trajectories. So they're never exactly sure where they're going to go. They line up a CT and the MR, and they're trying to target the SDN. They go down with a microelectrode and they record to find the bottom of the subthalamic nucleus. And these tracks go right by caudate nucleus, at least in this patient. Here's what we're going to do. We're going to do something called fast scan cyclic voltammetry. It's a method that was pioneered about 35 years ago. It takes advantage of the following. So I'm going to take I'll just do it in words first. I'm going to take a carbon fiber and I'm going to set it at different voltages. And at different voltages, different oxidizable species react at different rates. Okay? Everybody here take chemistry in college? Remember half-cell potentials in college? Let me see your hand. Come on, somebody took chemistry. There we go. Not a chemist. Okay. You know these charts, half-cell potentials. Well, these are reactions that generate free electrons, and so you have currents. And you can set a voltage, and the voltage can stop the reaction. That's called the half-cell potential, called the overvoltage. Okay? So that's what the idea was. Maybe we won't sit there and watch the reaction go to completion. We'll go whizzing by it at really fast rates, and we'll be able to get a signature for oxidizable species X and separate it from a signature for oxidizable species Y. It was really a maverick attempt 35 years ago, and they made it work. And it's easier to make it work using standard methods in a rat because you can take it and calibrate the electrode, go into the rat brain, and then calibrate it again. You cannot do that in a human being. When we manufacture the electrodes and they get sterilized, the first thing they touch is the human brain. That's it. They go into the sterile field, and they never come out again like that. So that was not available to us. And the other thing is these people have Parkinson's disease. Their dopamine is at least 100 to maybe 1,000-fold lower than the normal condition. Also, the whole field of cyclic voltammetry uses algorithms which, for this extraction of signals, are just not stable. I'll just give you an example here. So right here is the voltage sweep across 10 milliseconds. It's going from negative 6.6 volts to 1.4 volts, back down, and then we're going to wait 90 milliseconds. So the duty cycle is 100 milliseconds, and there's this witch's hat at the beginning of the voltage, and then waits, and that's 10 milliseconds. We're going to get one estimate of dopamine per witch's hat. What I'm showing you here is the current induced in the electronics based on that voltage sweep that's done there. And this little bump that's showing up here for bump, 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 this is in a flow cell, are different concentrations of dopamine. So we're increasing the concentrations of dopamine, and this bump goes up, and this counter bump over here that's not quite balanced in terms of area under the curve is the reduction peak. So you oxidize dopamine as you go up. You produce the O-quinone form. Some of it diffuses away. Some of it's still around. And when the voltage comes back down, it re-reduces on there. And that's what you see, the second bump here. A typical procedure, you put this thing in somebody's, rat's head, and you call some point in time 0, and you subtract that, this squiggly thing, and you produce background subtracted traces like that. In my mind, that's an illegitimate procedure. It's a linear subtraction of something that, if you just look at this curve, it's curving all over the place. Even as it comes off with concentrations here, it's not linear. Okay, so we wanted to throw that away. And we also wanted to throw away this 90 milliseconds because it was long. Now, I'm just going to show you a couple of things here because I realize I can go right down the wormhole here without focusing on the science. So I'm not going to focus on the new technology we've developed. I'm going to focus on the one that we did for these results I'm going to show you now. So here's the standard method that people use called principal components regression. What I'm showing here on the x-axis, this is time, and this is dopamine concentration, and this is in a flow set, so you have to bear with me. You have to get this technique in your head a little bit. These are 50-second steps of dopamine. What we wanted to do is tune these algorithms so if, as they're playing these games, if the dopamine sort of went up over the course of 20 seconds, 30 seconds, over a minute, that I could follow that. And then I also wanted to follow flash fluctuations. So this kind of step for 50 seconds, step for 50 seconds, step. This was me just picking a kind of temporal pattern in order to train the statistical model. This is on the same probe as the model that we're going to use, and I'm not going to go into this in detail. But there are lots of reasons principal component regression won't work on these data. We do something which is a modern machine learning technique invented by Zhu and Hasty, called the elastic net. I'm not going to go into details about the elastic net because I can't do it fast enough. I'll talk too much about it. Like I'm spare of words now, right? So as you can see, this is the same electrode, different algorithm. So everything here is the same except for the algorithm that extracts the signal. So we train up on some of the data, a model, a statistical model to predict the concentrations that we see in the flow cell, and we test on out-of-sample pristine data that we have that the model, that the elastic net has not seen. And you can see it's extremely stable. The other thing about PC regression is it gets tricked by pH changes, and that's an ongoing problem. In the literature where people use principal components regression, and I've drawn the elastic net algorithm up here, or at least the objective function, I actually don't think they get the answer wrong for the data they report. I think they have a very, very high false negative rate. I think they miss a lot of data. I think it's not stable and they have a false negative. Their false negative rate probably 80% or something like that. If somebody does this for a living, I'll entertain questions about that. So this is us training again the elastic net and the principal components regression on the same staircase steps here. You see that the blue stuff matches it. I'm showing the error functions here. Here's the error in nanomolar dopamine in a flow cell for the elastic net, and here's the error for the principal components regression. Let me show you one thing that I should point out. So what I've drawn here on this wiggly curve, so this is the wiggly current series induced by this voltage sweep, are the regression coefficients here. So blue and red are showing you spots on there that do well and contribute to the prediction of the dopamine concentration. And the white areas aren't. So what you can see is you got blue and red spread out over the whole duty cycle, not just at the oxidation peak for dopamine. So if you just say, I'm going to throw away this idea that I know the whole chemistry here, and I'm going to use all the information along there, it gives you a much better predictor, and it's stable in ways that the other algorithms aren't stable. I've drawn the oxidation peak as a band there, and whatever, peach, and then green is the other. And once you do that, you see a couple of things, and then I'm done with this method here, sorry. Let me draw your attention to G here. You see that you start seeing transients just randomly sitting there. Boom, we're sitting in the caudate nucleus. It receives a lot of dopamine projections. Even though they have Parkinson's disease, you can still see it, and you see things that even go longer, as you can see here. And when you compare it across the whole range of concentrations you might want to measure, what you see is that PC regression does quite well at higher concentrations, but it would do poorly if you were in a Parkinson's patient. It would do poorly at these low levels. Those are the reds versus the blues. All right, long story longer. I'm actually not going to apologize for going through that. That was a lot of us letting loose of assumptions and stumbling around, actually. And it's gotten even better than that. You can completely throw away the time structure of the voltage sweep and do a random burst. And mathematically, we now understand that. I won't go into that. This is one person that worked in Houston showing a very nice case of long-term timescale changes in dopamine, tracking an important value signal in this experiment. Let me just draw your attention to this plot right here. And notice there's a 1 sigma change. I've z-scored all this. 25 seconds. So this is a couple of minutes here. The blue trace here is the market. Basically, it's the price trace. And the black is dopamine. Now remember, this isn't just a quick thing. This is, makes a choice, the market goes up, he wins. Makes another choice, the market goes down, he wins. Loses, okay, so this is a slow change, but that's a 4 sigma change. Notice that as the market caved in, the dopamine, I'm not showing here, but the dopamine pulled out of the market. The other thing I haven't told you is that the markets that these people are playing, we used to do drift and diffuse models, and then I realized I had no principal reason to choose a particular drift piece or a diffusion piece, and so I've torn these markets out of the history books. That's the 1929 stock market crash, all right? And no one entered the 29 crash with more money than they left with. Nobody left with more money than they entered it with. It took everybody's money, including professional economists, none of which are in the room. All right, so when we look at the correlation over the entire set of markets and the decisions a person made, you can see that the dopamine correlated quite well with the value signal, with the market trace, if you do it within market. So much so that Paul Phillips said it doesn't correlate with button presses. We have button boxes that record actually pressure variations. We have to decide that they push the button in the task. You can say it doesn't correlate with these are button presses here. and this is the dopamine. But you can take the dopamine signal and you can give it to an artificial intelligence agent that sort of makes a bet depending on what the dopamine's done in the last 5 seconds. And it makes more money than the 354 people who had done the task at the time. Okay? The person playing it is right here, MH. He loses 20%. Okay? So as a value signal for this task, his dopamine is an expert and it's really good at it. Now, of course, he has Parkinson's disease, so it could be that the coupling between valuation and choice isn't so good in his brain, or it could be that you have a lot of other influences on the choices you make, and this is just communicating part of the information. So that's pretty interesting. Then we move to Virginia Tech, and I cold call every institution within a 2-hour radius and landed on these guys, the Wake Forest University neurosurgical team. It was led by Tom Ellis at the time. He's now since deceased. Steve Tatter is the lead here. It took about a year for us to re-engineer the electrode and fit into their procedure. This is a lot of work. And that transition correlated with us adapting to this elastic net regression method. So we went from getting 1 person in 3 years to we've now done 60 people in 27 months. So 60 people is a lot more data than 1 person. And it allows me to start looking at people on SSRIs, people that had different symptomatologies, et cetera. So we're quite excited about the effort. And we went there and we got to 17 people. And I divided out what does the dopamine do when I look at positive reward prediction errors and negative reward prediction errors, because I have this beautiful theory that accounts for all the data. That's what it does. It does nothing. Absolutely nothing. Okay? So I was very happy for about a week or 6 months. And then in the middle of it, Ken Kashida, the postdoc, that's the lead on this, who drove to Wake Forest, I think, 85 times, said, well, you know, have you ever considered this theory you wrote down, this whole fictive error piece accounts for it? And what if you sorted on the bets, you know? Because remember what I said? The foregone gain, as your bet gets higher and higher, gets smaller and smaller. So it bets all in. All you're looking at is pure. If you just believed it, and you believe that dopamine was carrying the signal, that's a pure reward prediction error signal. And I just took these data here, and I found every event, and there's a lot of events here, where I was bet all in, and voila, the positive prediction errors are encoded by positive changes in dopamine, and the negative prediction error encoded by negative changes. This is 100 milliseconds per point. We're now down to 4. I'm not going to show you any 4 millisecond data. All right, so that didn't feel so bad. So when we went back and dug up Michael Kahana's paper, Zaglul et al. In Science, and I guess that was in 2009, and they're recording in substantia nigra in human beings playing a game, it's called War, but whether you have unexpected gains and unexpected losses. And what you see is a spike density function here. It's aligned to the outcome at 0. This is unexpected gain. This is unexpected loss. There's an interesting offset between the loss signal and the gain signal, the error signal here. And this is what we see. I'm sorry about this. I constructed it right before I came. These are 100 milliseconds each point. And these are also z-scored, just like his spike density thing. So it's roughly equivalent. But it's transmitter. It's not spike changes. It's transmitter. So we have the spike changes on the left. We have transmitter on the right. But notice that the peaks are also shifted a bit. And we've done this again with higher resolution. And that's interesting. I can't explain that, but it's interesting that it corresponds so well to the only other experiment done in a human being, actually looking at the spikes in the nucleus that feeds the terminals there. Okay, but what about the bet dependence? Right? What about the bet dependence? So remember I said, this is what Ken came to me with. He goes, you know, if you just take the original hypothesis, it predicts everything we see. And latent in saying that the dopamine would encode the reward prediction error and the counterfactual information as a difference. There's a reason it ought to be a difference, too. I won't go into that. Then you have 3 predictions. The first is at high bets, dopamine encodes positive errors as a positive bump and negative errors as a negative bump. That would correspond to Schultz's data recording from the neurons themselves, and it would also correspond to the Kahana result in Science. But as the counterfactual piece got smaller and smaller, here it is right here. This is bet in 100% times the market minus my bet times the market. That's what I could have gotten here, minus what I did get. And if I factor out the market, it's minus R of t. Let me just do this here. You have this kind of minus R of t piece here. Remember, R is signed. You have 1 minus the bet. So as the bet approaches 1, this term approaches 0. But it gets bigger and bigger. It predicts that the positive, encoding the positive error should, as the bet gets lower and lower, it should sink down into 0, and it should invert. Completely unanticipated by any data in the world, just some stupid guess of this model. And the way we think about this is we actually think this is kind of like positive and negative valence pathways, but I'm taking too long. And so when you separate the bets that way, high bets, medium bets, and low bets for dopamine, you get exactly that. Okay? Now, maybe the model's wrong, but what it means is that this composite dopamine signal, at least in these patients, and this may have something to do with their pathology, I don't know, they encode prediction errors, but it compiles in with something like how good things could have been, but weren't because you didn't bet higher. So, for example, when you're at a low bet here, and you have a loss, or you have less than predicted, you have a negative error, it gives you this kind of relief response there. And the relief response would be just what you'd expect. I'm really glad I didn't bet any higher. So it's a contextualized version of it, now translated into dopamine. And you can't tell. You can't tell which dopamine molecules unless they're kept separate. You couldn't tell which dopamine molecules were carrying the counterfactual piece from those carrying the reward prediction piece. So that's an interesting finding. And the way that it relates to your emotional state, I don't know. But Parkinson's patients have a lot of affective difficulties. Many of them are on SSRIs, for example. All right, let me zip through. How am I doing on time? Everybody wants to go home? Am I done?
A few minutes.
A few minutes? I like that. In Australia, this is a few. Something about crossing the date line, right? You get an extra finger. Okay, this elastic net method lets us identify information that predicts dopamine concentration all along the trace. Let me just show you the picture. All right? So here's my little witch's hat again. This is 10 milliseconds on the x-axis on both of these. What I'm showing you on the y-axis is the time derivative of the induced current time series. Okay, so instead of just following it like this, I'm taking time derivative. And I'm showing you the way it responds in this little box here for these different color-coded concentrations of serotonin and for dopamine. Oh, here's another lesson for the students. Interdigitated but separable. Bad title. Way too many syllables. All right. You can see, though, these peaks are close to one another. If you were seeking to identify the dopamine peak and separate from the serotonin peak, it's almost hopeless. But the lines for any concentration wiggle differently. All you need is an algorithm that digs out the wiggles and doesn't just focus on the peaks. Okay? And that's what the elastic net does quite well. I can separate dopamine and serotonin. Here's dopamine concentration, serotonin concentration. I've trained the model. It can then predict dopamine and serotonin. This is a mixture model now. Stay with me. 3 minutes. 3 minutes is kind of exciting. Okay. So now we're going to go back into Ken Kishida's data, and we're going to match these trained models that we train in a flow cell to the electrodes that he had in the human brains, and we're now going to separate off serotonin and dopamine signals using this new method. Okay, and when we do that, and we do a median split over bets, remember at high bets you don't have a big counterfactual piece, and at low bets you do have a big counterfactual piece. And I'm showing you serotonin now. Serotonin encodes negative errors as a positive pulse. These are 100 milliseconds apart here. And the outcome is aligned to 0. I can talk about this negative 100 thing in a second. And here's a positive prediction error encoded by a negative going change in serotonin. These statistics aren't as great as they do when we repeated the experiment. And then at low bets, it's either gone to 0 or it's inverted, just like dopamine, but it's the other way around. So dopamine goes high on positive signals, serotonin goes low. Serotonin goes high on negative signals, dopamine goes low. It's the antipode to dopamine, but it's not quite temporally aligned. Okay? That's the first good recording of something that's been suspected in the literature for 20 years, and the person theorizing about it is Peter Dayan. So I've now become his worker bee after writing the theories down with him early on. All right? So as the worker bee of Peter Dayan, I'll go back and I'll ask, can the mixture model produce what I just showed you before for dopamine? And it does. It shows you it's not some mistake here. So I take this mixture model that can predict serotonin and dopamine, and I recapitulate Ken's results. Positive going dopamine pulse for positive errors at high bets, negative going results for negative bets, for negative errors. As the bet comes down, that inverts. And now here are the serotonin results just replotted on there. 93 seconds more, okay? Fine. What if we did a different kind of game now and repeated the experiment, but instead of just playing the investment game, I'm going to play the investment game beside somebody. I'll show you what I mean. This is what I mean. So I'm going to make a bet, and somebody else is playing in tandem with me. There's no consequence to this person what's going on. After I make the bet, 6 seconds later, the other person's bet is revealed to me. Okay? So I make a bet. I have a gain or loss. There's a tick, 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 tick. Pow. Comes either north of me or south of me. So I've either done better than the guy or worse than the guy. But there's no consequence to how much money I get or how much money they get. It's just a raw social comparison tandem experiment. All right? Then we do behavior on it, and we do bold imaging on it, and we look for signatures, and we can get a very good social comparison signature in the head of the caudate nucleus. So why not do this experiment in the Parkinson's patients? Again, trying to find out what computations are and aren't intact. And when you do that, we call it keeping up with the Joneses. Actually, that's a better start. And you see, I couldn't resist it here. So here's a colon, and here I am ruining everything about it. Right? Look at all those cells. I'm terrible. I'm still terrible. I'm 55 years old. I'm awful. All right. We also have them playing a computer partner. If we tell them they're playing a computer partner, you lose all these signals. Everything's the same. We actually replay human behavior on it, but we tell them it's a computer, you lose these signals. So they really do have at least some credibility as social comparison signals. You get a response in the ventral part of the head of the caudate. We can speed up the time scale here. Remember, it was seconds before on the first experiment, but this right here recapitulates what I just showed you. This is the serotonin signal on the x-axis. This is z-scored, on the y-axis. This is time on the x-axis here. Remember, these are 100 millisecond points. This is a kind of really nice, it's actually a double bump of serotonin here on negative errors and a downward deflection for positive errors. So that recapitulates result 1 on the 17 people. This is 6 new people. But at the social comparison here, this is the negative social comparison in green. This is the other guy was higher than me. You get a positive pulse of serotonin, and it ignores the other direction. Okay. I'm going to zip to the end and thank 2 people who helped me the most. Ken Kashida gets all the credit for all the heavy lifting here, and I picked the titles. So he can thank me for that later. And Rosalind Moran, who is no longer at Virginia Tech, she's at the University of Bristol. Thanks for having me.
It is time for a couple of questions, but I'm going to prerogative and ask the first question, which is largely to give you an extra 1 minute to expand on what you think is going on. So, you know, I have to confess I haven't recently read the original papers by with Peter Dayan. In terms of the positive prediction error and the dopamine and the prediction error in terms of everything you were talking about, that makes sense to me. But what do you think is going on with the serotonin? You didn't really explain that. You showed that it's showing this nice relationship.
Right.
But this is not something I'm as familiar with in the literature. Is this expected from a model that I'm not aware of? Or
There was no model of serotonin per se, but...
so why should serotonin be happy? Why should it be
there's a lot of diverse data that suggests that serotonin is about avoiding aversive things, waiting for events, anything to do with, let's call it aversion learning. Let's call dopamine prediction reward learning. Let's call serotonin aversion learning. The data are varied. They're at the wrong time scales. They're scant and whatnot. What this would mean is if you had 2 systems that all could do predictive learning, and the serotonin was carrying an aversive part, it carries 2 signs, positive and negative, and the dopamine carries 2 signs, positive and negative. And this provides the first evidence that you could support that. So you can have negative prediction errors with the positive system, with the reinforcing system. Things were less than you expected them to be. The aversion versus rewards is a statement of